Shai, can you please share
Yeah,
your
I'll
screen?
share my screen. Yeah.
Mhm. Okay, hello everyone. Welcome again for another session on spectroscopy today. As last time we discussed and uh, did the demo of the spectroscope machine, but we told you that we will be talking more about the basics of spectroscopy. So today, this is what we are going to do. Um, So let's begin with what is photosynthesis. It's a very basic concept. But before that, you feel free to open your microphone and ask any questions if you have any, or chip in if you have any interesting thoughts. For the time being, I'll just go on and I'll pose some questions to you as we go along this presentation. So, what is photosynthesis? We all know that it's a very basic concept. It happens all around us. This is where plants that have chlorophyll in it will eventually convert sunlight into food or glucose with the help of oxygen, carbon dioxide, and water around in the atmosphere. And then, as a byproduct, they produce oxygen in it. So plants have most of the plants that we see around us, including a lot of algae, have a pigment called chlorophyll in it. And at the very bottom of this hierarchy, structural hierarchy, chlorophyll is basically made up of chloroplast or these structural units, which are where you have these green plates stacked up against one another like a battery. So this is basically a cross section of a plant leaf where you can see the cells are lined up like bricks, and chlorophyll pigments, chloroplasts are basically within the cell that has chlorophyll, the pigment in it. Okay, next slide, please. So, what is algal bloom and why do we want to measure it? And what happens, when bloom happens? So, algal bloom basically is a significant environmental causes significant environmental damage. It can result in toxic effects on humans. If you have it in a water body, it can create imbalance in the ecosystem, whether it is a freshwater ecosystem or marine ecosystem. We all know that. But how do you measure bloom? Like last time, I probably said it in a single line that algae are of different kinds. Even the same genus of algae has like many species. Uh, so bloom can happen because of overgrowth of one species or five different species within the same water body. So how do you measure bloom? So one way to measure algal bloom is to not worry about exactly what kind of algae is in there, rather worry about how much chlorophyll do I see in the water body or how much of green do I see. So it acts as a proxy of how much green stuff is in there in the water body. Now, algae can be red as well, but most of the algae are green in color, which has chlorophyll. But the spectroscope machine that we will provide you, we are teaching you how to develop and do experiments with, can in principle identify different kinds of algae as well, because it's just light. Anything that has color absorbs or reflects light of specific wavelength. Nevertheless, uh, what are the current gold standard tools to do that? So the gold standard tools to measure algal bloom are satellite imaging or spectrophotometry or spectroscopy as we call it in short. So satellite imaging is hard. You need access to a satellite that is crossing over the lake and it takes images of the earth continuously. And once you get that image, you'll have to then process it, and it's not that accessible. And maybe on a cloudy day, you won't be able to see that lake, something. And as you all know, we don't have access to a satellite
And
image,
yeah, I
but
was
there
about
are
to say,
some.
sadly, most of us don't have access to satellites
Yeah,
through that we can
yeah,
control.
true. Unfortunately, I wish I had my own personal satellite, but no. But again, there are plenty of other issues as well. It's a ballpark understanding of where algae is more or less, and you need to have a lot of algae to be able to see it via satellite. Like for example, this first image is of, I think, Lake Erie. So Lake Erie has been having a lot of algal bloom issues, and people around Lake Erie know that. And the government has taken many initiatives while when there is an algal bloom, and they tell people not what to do and what not to do. So this is an image of Lake Erie. Then there is another image of a caldera, which is basically a large pond-like structure in El Salvador. There you can see that algae has caused a color change into a turquoise color rather than green in color. And there's another lake somewhere in India where I grew up. It's a small place called Silchar, and there you can see the pond has become red in color completely, indicating red algal bloom. So algal bloom, and by the way, people essentially in the last image, people can treat that water as drinking water as well. Obviously, they filter or they may not filter, but they use that water for washing clothes or maybe many other things. Um, so the problem is that you have blooms of different kinds, and how do you measure them? So spectrophotometry is one tool that can be used to do that, and it is also the gold standard. It's not that we are reinventing the wheel or we are starting a new technique that has to be validated. All we are trying to do is scale down the cost of doing spectrophotometry and making sure that the quality of the data is good enough. So, quick basic on the whole idea of generating spectrum from white light. You can watch these videos on YouTube, and we can send a few links as well. It's basically, I mean, if you type on YouTube how to split light using prism, you'll see that there are plenty of videos on it's a school experiment basically, that light comes in, you have a prism aligned at an angle, and then you can split light into all its components, at least the visible components that we can see. The other components we cannot see. So light primarily is an electromagnetic wave, and we only see a small part of that electromagnetic wave, which we call the visible spectrum, and it spans from violet-ish or ultraviolet, close to, I mean, far blue to far red. And in between, we have all the other colors. So all these colors are or wavelengths primarily is what makes our white light. So the next video is another example where, if you have a disk with all the 7 colors and you spin it really fast, you can make all the colors disappear, indicating that you can split white light into all the colors, and you can, make all the colors perceive to our eyes at white light if you run them really fast. So all these experiments that we teach in school these days were done by Sir Isaac Newton, maybe 500 years from now. So he was the pioneer in doing all that and trying to, see what light is made up of and all that. And he was also kind of crazy because he experimented with his own eyes. Apparently, I read somewhere that he used to insert a needle in his eyes and see if that changes his vision and all that. A true genius, essentially. So a quick question that I have for you all. What causes rainbow? Again, we all know that, but sometimes, these ideas or these thoughts, slides through everyday, chaos. But this is something you can think of. Maybe you all know the answer. I mean, maybe I can stop for a second and ask you what causes rainbows.
I always feel like I've never understood it, because all those droplets of water acting as prisms, why do I only see one spectrum? So I'd be very glad if you can explain that to me.
Exactly. So,
well, David, a related question is, why do we sometimes see two rainbows, a double rainbow?
Yeah.
this is not an easy answer. In fact, I have to I understand that the droplets act as prism, and then we split light. But how do they all align to make one large spectrum is something that involves reflection and refraction, as someone pointed out. But Dennis, two different layers of rain. Well, I don't know. So this is something I don't have much clarity on, like exactly. But maybe next time we can. Does anyone know that? Sai?
No, like I'm not sure. Like I guess I was thinking that as David said, that the droplets act like prism and they split the light. But yeah, I'm not sure why we see one arch that way. So that's something that can be our homework and figure it out. Yeah.
Yeah, we can come back to it another time.
Yeah.
Yeah, it's something
I
that
had also,
yeah.
yeah, I had also read that most people, when they look at a rainbow, really can only see about 5 colors. But the reason we teach children that there were 7 is that Newton thought there should be 7. He thought because it was a religious number, and that he probably didn't see 7 either, but he thought, well, they must be 7. And so you get these things like blue, indigo, violet, which are hard for most people. I can't distinguish them very well. I think I really see 5 colors, not 7. But Newton believed there ought to be 7 colors. And so now all over the world, we teach children to memorize the 7 colors of the rainbow.
Well, I mean, it's all perception, right? Because if you look at the wavelength, I mean, if I change, so red has a specific wavelength, right? And if I change the wavelength by, let's say, let's say by 50 nanometers. So red has a wavelength of what? On the order of 550, let's say, nanometers, right? So here it won't be, so red is, let's say, 650 nanometers. So if I move a little bit, I'm still red, right? But how do you define the boundaries of colors? Like somewhere between red and yellow is orangish, but like 20 nanometers back and forth, it can still be, I mean, you might call it orange. So I'm saying that it's all relative. So here you can see there are like more than 7 colors, but in general, we teach, you're right, students or schools children, that there are 7 colors, but it's all a gradient rather than well-defined colors. So I don't know why Newton came up with 7, but sometimes what mathematicians do is that they like symmetry, or they like to fit in, reality that based out of the numbers. And sometimes they say, well, my math tells me it has to be 7, but I can't see 7. So what's the question? I mean, back in Newton's days, there was no technology to basically go through wavelengths like that. He was just abstractly looking at splitting colors and checking them out. But coming back to perception, I would like to follow up with a bit of the technology that we all have in our eyes is the retina. And the retina essentially has cells that can detect colors. They're called photodetectors. There are 2 kinds of photodetectors that we have. One kind is called rod cells, and the other kinds are called cone cells. So cone cells are the ones that can detect different colors, like the primary colors. So there are pigments in the cone cells that will react to the 3 primary wavelengths, red, blue, and green. So if you have cone cells, if you don't have cone cells, then you are essentially colorblind. But the rod cells can detect light without any color. So we have a distribution of rod and cone cells in our retina to make sure that we see light, we see during the day, and also we partially see during the light. There is a difference in their excitability. When light falls on these cells, they get excited, and then they transmit an electrical signal that goes to your brain that we all perceive as light. This is where you can get a little philosophical as well, that in principle, we don't perceive light as light, we perceive electrical signals. Now, how does that electrical signal convert into red color what I'm trying to say is that your red may be a little different than my red, because we probably are not shooting electric signals at the same voltage. You know what I mean? But it's very hard to explain because there is no baseline here. However, one interesting thing that I realized is the the cone cells are less sensitive, uh, uh, and the rod cells are more sensitive, in the sense that, you know, when you want to see during the day and night, based on that, and then there is a difference in distribution of these cells in the retina as well, right? There is a blind spot we all have, where all the nerve bundles go back to the brain. That's where you have more of the cone cells, and the cone cells, and the rod cells, which do not have any color specificity but more sensitive, are distributed around the retina. So yeah, I mean, it's an interesting fact. This is how we have sort of evolved. Anyway, coming back to spectrophotometry, I have something called Beer-Lambert's law. It's a very simple concept to understand spectrophotometry. If I mean, during COVID, we all have measured oxygen levels in our body using this small, instrument which transmits light, and then there's a detector on the other side. Um, so the concept is very straightforward. If you have more hemoglobin in your bloodstream, that is a proxy for how much oxygen you have in your bloodstream. Hemoglobin absorbs light of a specific wavelength. In this case, it is infrared. So that's why, I mean, it's the borderline of red and infrared. So that's why most of these devices have a red color in it. The reason you want red color light to do this is because it has a larger wavelength, so it has longer penetration power. So it can go through your skin and a small piece of tissue like finger. So it can go through that. And on the other side, or on the same side, there will be a detector. It will just basically detect how much of that red light is transmitted and how much of it is absorbed. So the things that it can't detect and is essentially absorbed by hemoglobin, and that becomes a readout of how much hemoglobin you have in the fingers and how much oxygen you have, something similar. So our spectroscope is based on the exact same principle, where the absorption of light, or the absorbance of light, depends on the length of the sample and the concentration of stuff in the sample that can absorb light. And there's a mathematical constant E that we will not worry about at this time. That is a constant for the material. So a different material will absorb light differently. So let's assume that these beakers have water and the blue dots are algae. Okay, so if you're shining light, shining light with a torch in this example, which has an initial intensity of I naught, okay, and then after passing through the beaker with our hypothetical algae, the intensity is I1 in one case and I2 in the other case, okay, and the reason they are different is because we have different concentration of algae in the two beakers. The length of the beaker is same. The concentration is what changes here, right? So as you can see, absorbance will change. And the way we want to magnify this change is by taking a logarithm of the ratio of the two intensities, right? So if I divide the initial intensity I naught with the intensity at the exit, right, and take a log of it, I get a value of absorbance. So that's the calculation that goes behind giving you the value of how much algal bloom we have. And this is what exactly our device does. And that's a schematic of how we calculate algal bloom. Can we, yeah. So if you have a sample that has less algae in it versus higher amount of algae in it, so less green versus high green, and the green can be more or less like tiny microscopic algae, so it is suspended, so you cannot really understand what kind of algae, but the whole water sample is greenish, right? So the absorbance of light, particularly red light, will be higher in case of the high concentration of algae. And now you might ask this question, why are we talking about red light particularly? So there is this concept of complementarity. So if I place these colors in a circle, like in one of the previous slides I showed you, that if I place these colors in a circle, the red basically falls in opposite to, diametrically opposite to green. So in order to estimate the presence of anything that has green in it, the best wavelength to do that is red. So if you throw red light in the sample, green algae will absorb red light and then transmit the ones that it cannot absorb, and you will basically detect the transmitted and recalculate how much did I lose. You know what I mean? And another quick concept, green appears as green because it is not absorbing green light, it is reflecting green light primarily. So any color that you see, it is because it is not absorbing that wavelength, it is reflecting that wavelength. So that essentially means that the maximum absorption that stuff that looks green will do is of red. It will also absorb blue and yellow and all that, but we use just one wavelength for uniformity in our calculation. Okay, so this is a small experiment that I did where I took some water in a test tube and added blue dye, which not really blue, I mean on the borderline of blue and green. It's turquoise color. So there's dye 1, dye 2, and dye 3, as you can see. Dye 3 has the lowest concentration. Dye 2 and dye 1 has the highest concentration. Dye 1 has highest. So 1, 2, and 3 is the decreasing order of concentration. And there's another. There are 3 more tubes with real algae in them. So these are algae of one kind. They are called Chlorella vulgaris. They are microscopic 5-micron diameter algae. They form colonies or are mostly suspended in water. Every time you see greenish carpet layer on top of a lake or whatever, this is primarily Chlorella vulgaris. I had a purified population of Chlorella, so I just dissolved it in water. And it's an abundant algae, so you will end up seeing Chlorella, or their colonies. So I added Chlorella to these test tubes, and I have 3 different samples with decreasing order of concentration. So I did spectroscopy with all these, and as you can see, that the absorbance of red color in my samples decreases from sample 1 to sample 3 in case of both the dye, which is greenish, and algae, which is definitely green. And this calculation is fairly easy. And we can write a program for that once we get the spectrum from you, or we can have another session explaining what the backend calculation is. Building the spectroscope, I think we covered that. You need scissors, rulers, all that. And eventually your spectroscope should fit into your cell phone like that. And you should be able to see a spectrum like this, like a wide, can you go back? Yeah. Wide spectrum that has all the colors in it, and that goes from top to bottom. Okay. I think this we covered visible spectrum.
Yeah, I think this is covered.
Yeah.
So this is,
So these are few examples of spectrum that we got from the machines that we have been building. Okay, we have a quick question from Chip. Why use red light for red hemoglobin in the
that's supposed to be also.
Quickly. Blood is red.
I think you said it, but I needed to have
no. Yeah, I think it's a fantastic question. Blood, there's a conceptual difference here. Blood is red not because of hemoglobin. Blood is red because of red blood cells, which are reddish in color. Okay, so yeah.
So
hemoglobin,
hemoglobin doesn't give the red color to the blood, or does it?
No, I think that's more the red blood cells which do that.
Yeah. So the hemoglobin absorbs the infrared light and red light, And then the absorption is used as a proxy for oxygen. But the color that you're confusing the blood with is primarily because of red blood cells.
Okay.
Yeah.
So we have these horseshoe crabs called limulus here that have blue blood because it's, I understood it was a copper compound that carries the oxygen instead of iron. And that's why
Yeah,
their
no,
blood
I
is.
think, yeah, I think hemoglobin does have a role. It's not like hemoglobin does not cause it. I think the red blood cells are red because the hemoglobin has a role to play in that. But yeah, in this case, I think the red light is just a proxy for how much is being absorbed. I mean, hemoglobin will still absorb the red color. The red blood cells perhaps will have a different role, but the hemoglobin will perhaps absorb the red color. Now, I think it's interesting that why would
It
it?
just seems like if you're trying to measure green algae,
Yeah,
and you use red light, it seems like by analogy, if you were trying to measure red blood
that's a good
cells,
point.
you
Yeah,
use green light.
yeah. But one of the things I think, as Iqbal was saying, the red has higher penetrating power. The
Yeah,
red
that
light,
could be it.
which would also
Yeah.
useful. But yeah, that's an interesting question. That if the pulse photometer oximeter thing had green light in it, I wonder how much would have gone through, because it might absorb some.
Yeah, I think I need to
Yeah,
find out why
yeah.
we don't have. I think that the penetration is the answer. That it can penetrate. The red light is used essentially for this to go deep into skin and come back, and that's why they don't even use red light. It looks reddish, but they use far red, which is like infrared light. But yeah, I think efficiency wise, it may be better to use blue or green because of the complementarity principle. Okay, quickly coming back to this, we
I don't want to take it off too far, but just curious.
No,
no,
that's
no,
definitely
that's a good
interesting.
question.
We'll look at it. Yeah.
Yeah, we can come back to it. So spectrum is something like that, and as you can see that this spectrum has sharp lines of red and somewhere in the blue because it's a spectrum out of a CFL bulb. A CFL bulb has specific gas in it, so it can capture specific peaks in the spectrum in that way. And when you point your spectroscope in to a white light or to, let's say, sunlight, you will not get sharp lines like that, but you will get a spectrum as I showed you last time, just a general spectrum with all the colors in it, but that has information of how much algae is in it and all that. So in this spectrum particularly, if I draw a line across the spectrum and I look at the intensity of the wavelengths along this line, I will get a graph like that, where you can see that there is a peak at blue, there is another peak here, at the border of blue and green. There are two peaks in green, and then there's another peak in red. So these peaks have specific significance in terms of what elements are in there. So as you can see, that peak corresponds to europium, and then there is mercury, and then there's mercury again. So mercury essentially has two peaks. So if you have to detect mercury, you have to then have two peaks in blue and in green. So using a graph like that, which is called a spectrograph, we can detect presence of certain elements or presence of certain compounds in the sample. So that's what gives the peaks in a spectrum. So for chlorophyll, we have two kinds of chlorophyll at the molecular level. I didn't explain it before because we don't have to go into that detail. Chlorophyll A and chlorophyll B, they are just different configurations of the same molecule. And they absorb light at slightly different intensities, wavelengths. So as you can see, somewhere around 450, you have a peak of chlorophyll, and somewhere around 420 and about 470, you have two different peaks of chlorophyll. So this kind of spectrograph is what we are planning to develop as we get the spectrum from you. And also we are also experimenting with our devices here. I think, do we have any more questions on how the spectrum works? Yeah.
Iqbal, for this one, do we need to know the light source, the intensity of the light source we are using?
No, in this case, since we are taking two different spectrums, one is of the sample and the one is with regular water, we will basically do a ratio, so we don't need to know the intensity.
Yeah, like I guess what slide is kind of I should have added at the end is that, so which is why we're going to have one spectrum from water, one spectrum from the sample. So the idea is water will show you a spectrum which has certain colors in it, or mostly if you point it to white tile, it should have all of the colors in it. Now, when you add the sample and take the spectrum, there'll be some colors that will be missing from there, which will have been absorbed by the sample. And so we can compare these two spectra. The water essentially normalizes it and find out exactly how much color has been absorbed, which is missing in the spectrum. And that will be a proxy for the amount of chlorophyll, which is the proxy for the amount of algae.
Yeah.
Right.
Yep. Also, I just wanted to say something about the beer law. Can you go back to that slide? So essentially, the Beer-Lambert's law. Yeah. So essentially, what we are trying to measure is how much light goes in, how much light comes out. This log here is just for transformation of data into linear scale in a sense, because what happens is a lot of light can go in, a lot of light can get absorbed, but some little light comes out. So this is like on an exponential scale. So the idea is to minimize the differences between these two light points that we are measuring, the input light and the output light, and kind of normalizing it on the maximum scale. So basically the idea is just to measure these two light points, and log is just to transform the data. I just wanted to say that. And it can be any other transformation, but for Beer-Lambert's law, we use log 10.
So can you go back one slide?
Yeah.
So you've got mercury there with two different, twice, but with two different measurements. So how is that happening? You got mercury at 436 and at 546.
Yeah, that's a good question.
So
we have
so that essentially just means that mercury can light of both the intensities. I mean, you can see that both are not of the same scale. What I mean is that they are not on the y-axis. The peaks are not of the same height, right? So, but they do absorb light or in both
I'm guessing they just absorbs it for both the wavelengths.
So when you want to detect mercury, if you get just one peak, you can still detect it, but if you get both, then you can confirm that it is definitely mercury, something like.
Yeah.
But I don't know
I'm
why
guessing all of
exactly
these
mercury
have
does
like
it.
a signature, like all of these.
It's a signature, basically.
Yeah. So it doesn't have to be necessarily one peak. It can just be a signature type of thing.
Okay, thank you.
But yeah, we can go to, so David has made the spectroscopy, sent us some images. Iqbal, you can talk through them if you want. So I think these are the spectra that he got. And I think this is the idea. Like the spectroscope looks good to me.
Well, I mean, the spectroscope design-wise, it looks good. And David has essentially covered it up completely. But there are some problems. One is that there is stray light coming in from one of the corners, or where you have the.
I know what that is now because I've experimented a bit further. Okay. Basically, I don't have enough hands, so I'm steadying the spectroscope with one hand. I'm going to press the button to take a photograph.
Yeah.
But I also need to get the lens of the camera very tightly up against the diffraction grating.
Yep.
So, and light was leaking in. I tried to build a shroud that the camera would fit into out of Gorilla Tape. And there was actually that white light at the top of the picture in the middle. Yeah, that one.
Yeah, so that's the light that is coming in from the corner.
The shroud. I don't know what's driving the spectrum at the bottom right.
It's the reflection, yeah.
Yeah, so I think that would be the reflection on the camera.
Yeah, there are like more than one spectrums you can see, and it's not only 3 or 4. There are like multiples that we are not even seeing. It's because of that.
The sharp one is the one that I think is coming from that corner, and I mean, ideally you would want just one spectrum based on the light coming out of the slit, and it will be a little wide because the slit is, you know, because you cut out a small box where the slit fits in, right? So that's the width of the spectrum that you will get. But this is good practice, I think. The first time, yeah, I mean, you got that sharp line, and that indicates that the length of the device and everything is done nicely. You just have to find a way to connect your cell phone to the diffraction grating, like tape it there, like Foldscope, so that everything is close.
That's what I thought.
Yeah.
What about the diffraction grating? Have I got it the right way up?
Yeah, the diffraction grating is perfectly aligned, I think. Yeah, because that rectangle should fit into the rectangle of the device, right?
Yeah.
And I think just for like future, what will happen is once we have more sharp spectrums here, so we're going to take on our back end, you can write a program to do this in the future. We're going to take the tap water in daylight as the normalized sample, and then in this case, the pale pink liquid that he found, and then compare these two spectrum, and there will be some bands in this pale pink sample missing, which is there in the tap water. And that way you can compare them and find out exactly what wavelength was absorbed, and then try to come to a conclusion as to what there was which absorbed that band. That's the analysis that we will do on the back end once we have a lot of these data coming in.
Yeah. Can I ask another question? Looking at the samples I've got, quite a number have macro amounts of green material in them, presumably algae, but might be some vegetation, plant vegetation. Do we need to sieve the samples in any way before putting them in the spectrophotometer?
That's a great question. So if you have a sample that has a lot of leafy green structures, then I would recommend just filtering them once, instead of, because they will block light, because we are trying to detect algal bloom because of algae that is primarily microscopic rather than leafy structure. So leafy structures are great for microscopy, so you can image them. But if you end up having a lot of large chunk of leafy structure in the cuvette, then that will be, that will block light in some way.
Yeah. I think Dennis has a question. How do we filter?
Yes.
Yeah.
There are like so many ways. I'm trying to think of one good way that is easier to use.
There's various tools in the Foldscope thing. Looks like pieces of fabric with a particular.
Right. Yeah.
I don't know whether they're too fine.
So one of the Foldscope box that they sell has filtration equipment as well. So they sell these different kinds of meshes of different sizes, and then you can use them to filter something through a cup. The other ways are, so there is something called painter's filter. So it's a piece of paper cone that comes in with a filter at the end. They filter paint using that paper cone. It's very cheap, easily available online.
I think also coffee filters.
Yeah, coffee filters might also work. Yeah.
Coffee filters may not work because it's again paper. Will it let, let's say, 5 micron, 10 micron stuff go through? You know what I mean?
And maybe a piece of fabric. Small piece
a
of
piece
cloth.
of fabric or strainer will also work. All we want to get rid of are the thick leafy stuff. The other thing that you can do is, if you have a sample in a bottle of water, let's say, and you have a lot of leaves in it, they will float on top, and then you can get rid of them and then take the sediment and shake it up. So that's the microscopic algae that you have in your sample. So the leafy stuff can be physically separated. I would leave the bottle for some time, or you can use any strainer that you can find off the shelf. There are like tea strainers and so on and so forth.
Yeah. Another question is that, so won't we need fresh equipment each time? Otherwise, we will cross-contaminate. I think
You
like
can just
as long
wash
as
the
it's- yeah.
cuvettes, I think. Yeah,
Yeah.
just wash the
Yeah.
cuvettes with regular water, and then I mean, in the packages that we gave you, I think you have at least 4 cuvettes. Just wash the cuvettes, and it should be
Yeah.
fine.
So okay, he says he means the filters. So yeah, the problem with filtration is that this is an active problem, right? There is no clear answer as to do this and you will get filtered. So what we try is we can use small pieces of cloth or some other filter that you can purchase. But yeah, that is kind of an active experiment that in general people are doing to try to figure out what's the best filtration method. There is no one clear direct answer as to what the best method would be. It's
Yeah,
something
I
that we
mean,
are also continually exploring.
so on Amazon, you can buy this. If you type painter's filter, these are paper cone filters. They will cost you around $5, and you will get about 100 pieces out of that. So you can reuse them. Sorry, you don't have to reuse them. Yeah, I'm just looking at it right now. Maybe we can send you a link. Is that something that we can do?
Yeah, we can also send this link.
I mean, that's a good alternative to paper cloth and all that.
Okay, and I think Chip also sent these
Yeah,
images in of
we
Foldscope.
will quickly discuss Chip's data upload that he sent us recently. That's a beautiful hat there. So the alignment and all, I think, looks great. We can clearly see the cells on the left image, and I think, Chip, can you just explain to us like what are the right images on the right are?
Yeah, sorry. I think the second one was onion skin.
Ah.
I mean, not onion, garlic. From a garlic clove, I just got some of the thin skin around it. And the third one was from some pond water. We actually have right where I live, many of our ponds, I think, are oligotrophic. They don't have a lot of stuff growing in them, which in a way is good, but it's not so good for the project. There are ponds on the Cape that have a lot of algae, and it's a serious problem. So what I need to do is figure out which ponds to focus on. Maybe one right in my neighborhood where they seem fairly clean, and then another one that's more algae. But this was from one of the cleaner ponds, and I don't know what I'm looking at. That was the best picture I could get.
Yeah, sometimes, when you have microscopic algae, they can get lost in your sample, and you have to find a way to, or maybe try to scrape some algae and try to do microscopy with them. Like some of the algae you can see physically attached to a stone or, somewhere there. You can scrape them up and try to make a sample out of it. This is just practice. So try to see if you can make good samples out of those algae that are definitely algae attached to a stone and all that. Moss, yeah.
Yeah, what do you think these lines are? It looks like a little triangle and some other lines. What would that be?
Yeah, these lines, I think, are lines on the plastic or glass, either the glass slide or the plastic cover on top of the ring stickers. So I think you were focusing on the plastic rather, and maybe there's nothing in the water across. So you were like trying to focus and you find some patterns and you image that.
Yeah.
So yeah,
Okay, well that helps. I just need to do more.
I don't think these lines are active matter, like living things.
Yeah.
I had one point. Can you go back to David's slide with the spectrophotometer? So here, so these pictures, so I think there is also a lot of sunlight from where you are taking the picture, right? So I think this was done, this was not done in a room or something. So I think, will it not be better to do it in a room with a single light source and pointing the spectroscope towards it, right? Because if there's a lot of light even coming from the front side, you will not be able to take clear pictures. Here, the problem is not only multiple the bandwidths that we are seeing, but also a lot of light on the front end. So I think it would be better to do it indoors instead of outdoors, right?
Yeah, I was thinking of trying the little illuminator you get with the Foldscope. and switching all the lights off to get a dark room. It makes the handling problems a little bit more severe, but
Yeah.
Or even a bulb in your room and you can just point it towards it. I'm just saying that there will be less like source of light that can get into your spectroscope. So that will give you a clearer image, I think.
Yeah, I think that's
I mean, try light with an indoor lamp that is consistent, because that will stay there with more or less the same intensity for many months.
Right.
Compared to outside. But I mean, once you have the box sealed properly, there's only one way light can enter into the chamber. Once you have it sealed properly with the slit, and on the other side with your phone, then you will get only one spectrum. And the fact that you are checking the spectrum, taking an image of the spectrum for both water and algae, we can do the ratio. So I mean, incident light should not have that much effect, I think.
Yeah, in that context, another comment that David had in the chat and Dennis had in the chat was that also, like as long as we do have the water to normalize it with. So if there are slight errors and things like that in the water sample, when we take a ratio, those things do get normalized. So that part helps.
Yeah, I think what Dennis is suggesting is if we use the same filter, then it also eliminates another variable, and we can compare data of from one person to another person. Yeah, that is true. Yeah, I mean, we'll send you a few links of those filters that you can find on Amazon. There, I mean, if you want, you can buy it, or you can find your own ways of filtering them. Because if you have a coarse enough mesh, most of the algae will pass through, except for the leafy stuff, which we don't want anyway. Yeah, for the spectroscopy. Yeah.
Okay, I think for time, I'm just going to quickly show the data upload part. And then just in future, we will have another spectroscopy building session. These filtration problems, and also we'll try to address there. And probably not next week, but in weeks after that, we'll do another demonstration. We can do more troubleshooting of the full scope as well. And we'll definitely have another spectroscope building class where we can just work on these things in detail. For now, I'd like just to show you what you can do with the images that you take. For now, you've been sending it over via email, but what we have essentially on our websites, we have created dedicated portals for each of you, where you can now log in and just submit your images there. What it does, it serves two purposes. One is you don't have to keep emailing back and forth. We get to see the images on the back end as well, as and when you upload. And second, it creates essentially your own lab notebook type of structure, where you can log in every time and just get your own images. So I'm going to go through the instructions on how to create your portal. The meeting's been recorded, and I'll send a detailed instruction list as well. So if you go to our main website, that's openfieldcollective.org, and you click on the database icon on the right. If you click here, then it takes you to the database part. You'll see if you scroll down, it says choose upload database. There are many different options here. For this cohort, you're going to click on this, the NewScientists. We have other cohorts active as well, and the other tabs are for them. So you can just scroll down and see some of the images that other people have uploaded as well. But if you click on NewScientist, it'll take you to the NewScientist algal bloom database. When you use it for the first time, you're not going to have a username password. So like here on out, it's pretty self-explanatory, but I'll go through the steps. So it says if you do not have an account, you essentially click here to register. If you click on that, they'll ask you for an email and for you to choose a password. So what I've done is all the emails that we have been communicating on in the OFC email, I've set those emails already in the system. So when you type your own email and then choose a password and you hit submit, you'll get an instruction that you have received a verification link in your email. It should be pretty self-explanatory once you type it in. But just so that if you say use a random email that we have not communicated on before, it'll say account not ready for creation. So when you do sign in, just use the email that we have been communicating on in the OFC email. So once you type your email in, choose your password and hit submit, it's going to send a verification link to your email. You click on that, then your account gets activated. Once that is done, you can just go to the login here. Again, come to the NewScientist algal bloom database. Whatever username you have, the username is essentially the email. You put your email here and the password that you chose. I'm going to log in with my database. So once you log in, you're going to see something like this. So once your account is created, it'll provide your email, and then this is kind of what your portal will look like. Okay. First, there's this section about so there are 3 sections: water body details, algal images, and spectroscopy images. First is the water body details. You don't necessarily have to submit something here, but say you want to essentially document what lake you're observing, and if there's any interesting appearance that you want, say you can. So whenever you collect sample, you can say I'm collecting on the 4th, and you type whatever the name of your lake is. You can type your location. In my case, it's San Diego. I'm just going to put that. If there's any specific algal appearance that you want to put in, whatever color you're seeing, whatever odor you're getting, pH is not a required field, but if you have that, if you have a measurement of that, you can put that in as well. So I'm just going to put in test for now. We'll put in green here, and here I'll put test as well. You don't have to submit pH. And if you hit submit, essentially you can see that it gets stored date-wise here, so that like in future you can go back in and say, okay, on this date, this is what the sample kind of look like.
Yeah, I just want to mention. So yeah, like a lot of people or some of you wanted to do multiple lakes. So now it is very important here that first you keep the lake names consistent and you describe the lake name here correctly.
Yeah, that would be helpful for us, because when we get the data, we sort by lake. So in this case, suppose you forget to give the space next time, so that kind of creates an issue that when now our system might classify this as 2 different lakes, because in one case you forgot to give a space, which is fine. It's not a problem. Just I don't have editing or deletion power here. So if there's something you want edited, just email us and I can manually go in and edit and or delete your entry for you. So that's the water body details part. This is the lesser important part of things. If you click on algal images or just scroll down, it'll take you here. This is where you can, for now, keep uploading the full scope images that you're getting. So essentially, it's again self-explanatory. You choose a file. I'm going to choose a file. I don't think you guys can see me choosing a file, but so you choose a file. You add an image name, whatever you think you're seeing, full scope image of whatever, and then you can add a description as to what you think you're seeing or what lake you chose it from, taken from lake A, seeing algal sample, whatever you want us to know. And then if you hit upload, it's going to say image submitted. And then what happens is your images get stored, keep getting stored, all the images that you've taken so far. If you click on see gallery, it shows you all the images that you have submitted so far with the descriptions that you have added. Right, so it kind of maintains your own gallery also of images that you have always taken. And you can now hit go back to database.
So yeah,
and come
I'll
back.
also just suggest one more thing. So like image name or image description. I think if you remember to add the lake name and the date, it will be helpful for you and us as well. So yeah, just those tags will be really helpful.
Like as much description and image name as you think like would help us to know what exactly the image you took was. That definitely would be helpful. Do we have a question in the chat? Okay, that's for Silja. Okay. So this is for the algal images. I think everyone can start to at least put the full scope images here. We'll get a chance to see them and figure out how the full scope is working. When we get the spectroscope on, this method is again the same. You go to your date of collection, you choose your date. Now here you have to first choose the water sample file. Right, you can write some notes here as to what you took. Then you have to choose this spectrum with the sample in it, right? Whatever description you want, and then hit upload. And again, similarly, both the water and sample images at every date will keep on getting stored here. And if you click the gallery, so these are images that we have taken before on May 4th of this is the spectrum that I got with water. This is the spectrum that I got with the sample, or more accurately, Iqbal got with the water and sample. So again, like as you keep on uploading spectroscopy images, they're going to keep getting stored here as well. So that is kind of the idea. First time users click on get a new account. You use your email to set your email and password. Once you log in, this is essentially what your lab notebook would look like. And you can keep storing your full scope images here and your spectroscopy images and any other water body details that you might want to provide. This saves us the trouble for not having to email back and forth. That email thread is now reaching 100. So it saves us the trouble. You can just put your images here, you guys get your own storage database as well. And on the back end, we can analyze and see the images from here as well. I'm going to send out a detailed instruction as to what I said, the step-by-step. That should also help. And you guys have the recording here as well. I'm going to pause if there are any questions.
I just want to say that you can still send us emails. Though, I mean, you can upload your pictures there, and if you want to discuss something, you can still send us emails. We are just trying to reduce our storage space on the email that we have. So that'll be good.
Dennis has a question that can it be a river? Can he take algal samples from a river? Iqbal?
Yeah, sure. I mean, you can take it from any water body, but rivers usually have a flowing, I mean, rivers flow. As a result, the algae is always moving forward if there is algae. So it may be hard to check the stagnant bloom that is going on, but I'm sure rivers also have algal bloom going on. So you can check. I mean, I don't see there is a problem with that.
Yeah, and regarding the filtration question, definitely we're going to send some more filtration.
I've
directions.
shared a link here on Amazon, but this is Amazon.com rather than the UK link. But the product should be similar. We will send one from the UK with the UK link, David. Yeah, and I was just, as Shaik was explaining the data upload part, I was just trying to figure out Chip's question on hemoglobin. So I think the answer is primarily penetration. So it turns out hemoglobin has two different configuration. One is oxygenated when the oxygen is bound to it, and the other is non-oxygenated when oxygen is not bound to it. It has delivered the oxygen to the cells that it was, targeting. So the bounded state where the oxygen is like loaded onto the hemoglobin molecule, it absorbs more of the infrared light and lets go of the red light. It doesn't absorb that much. And I think red light is used, I mean, when you look at a pulse oximeter, it has both infrared and red light, I think. But red light is used because it can penetrate the skin and the tissue, and then it can go and define the detector where you can now see how much of red light did I get, in each heartbeat. So that will give you an indication of how much oxygen is the hemoglobin carrying. So that is the whole idea. But you are right. The blood is red because of hemoglobin in it, because hemoglobin is what fills the red blood cells, and then we call them red blood cells because of hemoglobin. True.
Okay, I think for time we can.
Okay, so please try to make the spectroscope device and send us images as Ankit
Well, can you
mentioned.
send the UK link for the ring stickers? I think this is what you're asking for, David. Is right?
Yeah,
I'm
using those fairly quickly, and they do work really well. So it'd be nice to get more.
Let me note that down. I can find a way to
the filtration
connect with
links,
some vendors there. Yeah,
filtration links and the
ring sticker for UK.
Okay, and and please try to make your own accounts. If there are any difficulty with that, just send us an email and I'll look into it. And then keep uploading your images onto the portal. And we'll organize something else later where we want to troubleshoot and demonstrate again. Okay, I think